All right, so today we are going to finish up metabolism, which is basically how the body handles what the digestive system does, which is why we typically cover it right after the digestive system. And it does kind of take care of some of the digestive system stuff that gets left unfinished if we just stop at things getting absorbed. So we left off with gluconeogenesis yesterday, and now we're going to tackle lipid metabolism, then we'll do proteins, and then we'll talk a bit about the liver. So for lipids, we are going to pick up where we left off, talking about how they have some special features for getting them digested and absorbed, because they have to be emulsified by the bile salts to give the enzymes access to digest them. They then get absorbed as micelles, packaged up into the enterocytes. Now, once they get into the enterocytes, they actually take the monoglycerides and the fatty acids and they make triglycerides again. So immediately we're right back to triglycerides. And again, this is too big to get into the blood. So instead of just kind of diffusing into the bloodstream, which is what happens to every other nutrient, all of our lipids and lipid-soluble substances continue to get packaged for transportation. So the packaging that the enterocytes do becomes something called a chylomicron. So this is a combination of the triglycerides, cholesterol, phospholipids, and any other lipid-soluble things absorbed from the meal. It gets exocytosed out of the cell and gets absorbed by the lymphatic capillaries. So the lymphatic capillaries within the small intestine are called lacteals. Lymphatic capillaries are able to take up larger things that can fit into blood capillaries, so that's why that's what's doing the job. And the lymphatic vessels eventually empty into the circulatory system. So they do end up getting into the blood and then circulating with all the other nutrients, but they have to take this extra pathway first. And they have to be in this special packaging. So the chylomicrons are, again, just like the micelles, they're all these nice bundles where the phospholipids are on the outside. There are these special proteins called apolipoproteins that kind of tell the body what this little package is and guide it to where it needs to go. And then all of our triglycerides and other things are on the inside, safely away from all of the water on the outside. So the chylomicrons typically get processed by the liver. So as the blood circulates through the liver picks up the chylomicrons and processes all of the different lipid-soluble substances. Adipose tissue cells can also pull the triglycerides out of the chylomicrons and store them. Just depends on the body's needs at the moment, which we'll address in a little bit. That's not the end of the journey though. So the liver is the first step in processing, but the rest of the body has need for triglycerides, especially cholesterol, because cholesterol is, I don't know if you remember this from when we talked about it at the beginning of the class, but it's part of every single cell membrane, and it gets converted into a variety of steroid hormones. So we use cholesterol for a lot of different things, and so most of the body's going to need some, and this is where they're going to get it from. The liver is going to send it out and let anybody who needs these substances pick them up. So we use carrier proteins, specifically lipoproteins, and we bundle them up, again, into a little package. Now, instead of the package being a chylomicron, it's what we call a VLDL. So VLDL is very low density lipoproteins. It's low density because it's got lots and lots of triglycerides in it, as well as some cholesterol. So VLDLs get sent out into the blood by the liver, and anybody who needs triglycerides will remove them from these guys. Most of the time we're going straight to the adipose tissue for storage, but not necessarily. And the lipoproteins continue to circulate in the blood. Once a certain amount of the triglycerides are removed, they become LDLs, or low density lipoproteins. Now they are lower density because they're a lower proportion of triglycerides. And so this becomes one of the things that we measure when we're looking at people's cholesterol levels. So we've got low density lipoproteins as a marker of the amount of cholesterol circulating. This is also considered the bad cholesterol, because when these circulate in the blood, sometimes they can get stuck and they can form partial blockages and damage blood vessel walls. So this is actually why cholesterol in your diet has no effect on your bad cholesterol levels. You can't really control your cholesterol levels by controlling your dietary cholesterol, but you can control to a certain extent. There are genetic components to this, but you can control your cholesterol levels to some extent by controlling the amount of triglycerides in your diet. Because the more triglycerides you consume, the more VLDLs get sent out, and the more LDLs are there as the byproduct of the VLDLs taking the triglycerides to the adipose for storage. So the LDLs can be removed from circulation by any cell needing cholesterol. And then the last type, the last kind of branch in this whole lipid transportation chain is HDL. So HDL is high density lipoproteins. And that's because HDLs are mostly just the lipoprotein shell. The liver makes them and sends them out into circulation without any lipids in them, so that any tissues that have excess cholesterol or excess phospholipids can basically make a deposit. And then the liver removes the HDLs from circulation and secretes excessive cholesterol into the bile. So this actually takes excess cholesterol out of circulation and helps you keep your keep the cholesterol out of the places where it can cause harm. And so this is why the other component of what we measure in people when we're worrying about cholesterol is HDLs. So this is, the LDLs are what some people will call the bad cholesterol, because this is the form in which it's more likely to cause problems in blood vessels. And then the HDLs are what people call the good cholesterol, because this is how the liver is kind of cleaning house and getting rid of excess. And neither one of those is necessarily true. They're all part of the same pathway, but certainly we see associations, negative associations with people with high LDL levels and better associations with people with high HDL levels. So a couple of diagrams to show you that. So this one is kind of showing you parts of the pathway, the liver secreting bile into the intestines reabsorbing some of it as chylomicrons, and then VLDLs being formed. And then there are other things involved here that I did not cover, and I don't want you to worry about it. And then it's kind of showing you the HDLs. This particular one is dealing with the various types of apolipoproteins, apolipoproteins. This one I think is probably a little bit better as far as kind of showing you some of this stuff. So this is showing you a couple of different ones. So we've got like chylomicron formation from absorbed micelles, and then they're kind of skipping some steps here. Oh, here it is, sorry. And then the chylomicrons go out, and then it's kind of showing you the link there. Anyway, I found a couple, and they look pretty. This one's probably a little more useful, but all you need to know is what's on the slide. So basically, lipids need escorts when they go through the blood, and those escorts are lipoproteins. And the liver is the major player in dealing with what's circulating. Now, when we need triglycerides, because really triglycerides are just stored energy. They're the most efficient way to store chemical energy trapped in chemical bonds. And when we need to use them, we need to undergo the process of lipolysis. So lipolysis is where we actually break down the triglycerides and we use those fatty acids and glycerols to make ATP. So we call that fatty acid oxidation, and it's basically how we can insert fatty acids into the cellular respiration pathway that we talked about last time. If we're using a lot of triglycerides for fuel, we can sometimes get a buildup of the precursors more than we can actually use. And so at that point, we will undergo a process called ketogenesis, which is where we take acetyl-CoA, which is the form of carbon that we send into the TCA cycle. We'll convert that into something called ketone bodies. So ketone bodies circulate in the blood, and they can be used by different types of tissues to make ATP. So this is kind of the liver's alternative to releasing blood glucose or to releasing glucose into the blood. Typically, it only happens in times when there aren't enough precursors that can actually be made into glucose, because this is more like a backup pathway for starvation. Now, usually I don't talk about that, but ever since the keto diet became popular, I feel like we need to discuss it a little bit, because this is the basis for the ketogenic diet. So the other term for fatty acid oxidation is beta oxidation. They mean the exact same thing. And this is the chart in the book. Oh goodness, sorry. Sorry. So this is the flow chart in the book showing you how that works. Glycerols are really easy. Glycerols can actually go, oh no, glycerols can go through glycolysis. They kind of enter like halfway through, and they just kind of go through the whole thing. It's like having half a glucose. The fatty acids can't be used in glycolysis, but with some modifications, which is where the term beta oxidation comes from, they can still be rendered into acetyl-CoAs. Basically, fatty acids are just chains of carbons, and acetyl-CoA is a 2-carbon chain with this extra thing on it. So we just kind of snip this chain of carbons into 2-carbon chains and feed those into citric acid cycle, and then they go contribute a bunch of stuff to the electron transport chain. And then this is the flow showing you the alternative pathway. So if we have more acetyl-CoAs than we actually need, and we're not wanting to run them all through the pathway, we can't turn it around and turn it into glucose, but we can turn them into ketone bodies, which beta hydroxybutyrate and acetoacetate are 2 of the major ketone bodies. And like I said, then the liver would release them into circulation as an alternative fuel to glucose. So even tissues that are wholly reliant on glucose can convert to using ketone bodies if glucose is limited. And that includes the brain, where typically neurons can only use glucose. Like we're talking about the liver using all sorts of different things, and a lot of other tissues do the same thing, but neurons are pretty specific. They need glucose and only glucose. So if blood glucose levels are low for prolonged periods of time because of a lack of carbohydrates, then they will switch over to using ketones instead. And basically they will take them and run them through the TCA cycle. So usually the only time this happens is during prolonged starvation, which is probably what we, why this pathway exists evolutionarily. And then we can also see it in uncontrolled diabetes, because in diabetics, the body thinks it doesn't have any glucose, it thinks it's starving, and so it switches over to this instead. The excess ketones will get converted back to acetone and carbon dioxide in an attempt to get rid of them. And this will cause acid buildup in the blood. And then the acetone will actually be kind of secreted in various substances, and it'll make people smell kind of sweet. So this is just kind of showing these guys. So here's the ketone bodies, and we can, again, put them back into the citric acid, aka Krebs cycle, and then it'll go through electron transport and we'll get ATP from it. So this is probably the most efficient way that we can use lipids. And we do a certain amount of this all the time. And we'll kind of look at when that's more likely to happen in a little bit. We can also make more lipids. So dietary triglycerides aren't the only source of triglycerides that are in our bodies. And that's because anytime we get acetyl-CoA, we can just put a bunch of them together and convert it into fat. So even when we have a lot of carbohydrates or even amino acids, using them as fuel, if we have more fuel than we need at the time, we just convert it to fat for storage later. Both adipocytes and hepatocytes can do this, and it happens pretty readily whenever we have more nutrients available than we're using. So obviously, which thing we convert these guys into depends on the need. So when we don't need more fuel molecules, we just convert it all into triglycerides in this process of lipogenesis. If we need more fuel molecules, then the liver will convert it into ketone bodies because it doesn't have enough stuff to convert into glucose. And that is, don't be intimidated. You don't need to know any of those structures. It's just kind of showing you the beginning of the process of making triglycerides by showing you how we can chain acetyl-CoAs together to make fatty acids. That's all. This is a less intimate, maybe, I don't know. This is showing you a flow of sort of the movement of lipids through the liver. It includes lipogenesis, which is what I was looking for when I was looking for a diagram, even though they spelled secretion wrong. That's cool. Cool. So this is showing you, for example, we're taking glucose through cellular respiration in a liver cell, and we can break down fatty acids and send it through the pathway as well. But if we generate excessive acetyl-CoA and we don't need to use it for fuel, then we can make it into fatty acids, put them together into triglycerides, and send them off to the rest of the body in VLDLs mostly for the adipocytes to store. If you're curious about the keto diet, here are a couple of articles reviewing it. There's mixed stuff, and it hasn't been around for long enough for us to know if there are long-term consequences to the diet. The really short version is that it works for some people. It's really hard to stick to because you have to keep your carbohydrate intake highly limited, and it can actually be hard to meet your calorie requirements because you have to make so many of them from fat. So what we know about diets is that if you're doing something that takes weight off fast, but it's not a sustainable diet for the long term, that you don't keep the weight off. And so to me, that means for most people, this isn't the right diet, because if you can't sustain it, then you're just gonna like lose weight really fast, be happy with it, decide you want to eat carbs again because they're awesome, and then you gain the weight back and you're bummed out. But it does have a place. You should always do it under the supervision of a doctor, though, because it is kind of tricky to do. So if that's something that you or anybody you know is curious about, here are a couple of decent links about it. Which takes us to proteins. So protein metabolism. So we talked about how we are mostly active transporting amino acids or absorbing them by other means into the enterocytes. So now we're going to look at what happens after they end up in circulation. So the difference between protein metabolism and carbs and lipids is that we don't store protein. We don't have an equivalent of triglycerides or, yeah, I just totally lost the storage form of carbohydrates. It'll come back to me later. We don't store them. We use the amino acids to make new proteins. If we don't use them for proteins, then we get rid of them. So when we have excess amino acids from a meal, we can either use them for energy, or we can convert them through the pathway and then use them and then kind of just make fat out of them, basically. In order to use them for energy, we have to remove the amine group, which is the part that has nitrogen in it. And then depending on the amino acid, it can either get converted to pyruvic acid, which means you can make glucose, or you can throw it into cellular respiration, or one of the molecules in the Krebs cycle, and then it goes through the Krebs cycle. This came out even blurrier than I thought it would be. You don't need to know any of this. This is just showing you where different amino acids can be placed into cellular respiration, specifically into the Krebs cycle, although this is also showing you that anybody that can go in here can be made into glucose. So these are gluconeogenic amino acids. Everybody else can at least be burned for ATP. So protein synthesis then is the major anabolic process, and what we're synthesizing just depends on the body's needs. Because protein makes up such a massive amount of our bodies, amino acids are actually the most important anabolic nutrients. We can make do with carbohydrates and fatty acids, like we can sort it out, but we need all of our amino acids to make all of our proteins. That being said, when we aren't using all of our amino acids, we're generating a lot of nitrogenous waste. So all of those amine groups that get removed with those nitrogens on it, we have to get rid of them, because otherwise they're going to create toxicity issues. And so there's two things we can do to them. Transamination is where you transfer them and make a different amino acid out of it. And so that way we don't have a waste product. We're just kind of modifying something else and getting something else we need. But if we truly have an excess, then that's going to mean we have to remove the amine group, which is called deamination. And then we have an ammonium as a waste product. Ammonium is NH4+. It's like a weak acid. And so we have to get rid of it. And in order to get rid of it, we do something called, we convert it into a molecule called urea. And urea is the primary waste product we have for nitrogenous wastes. The kidneys eliminate it for us, but it's not as simple as that. And so because of that, we have something called the urea cycle. So with the urea cycle, it's just kind of this crazy thing where we can kind of cycle through various molecules in order to ultimately get urea as a waste product out of the ammonium. So it's not as easy as just converting it. We have to send it through this complex series of reactions. Now, we don't need to know them. I just need you to be aware of the fact that the ammoniums are coming off of the amino acids when we use them for fuel. We have to convert that into urea in order to get rid of it. And then we're going to take that urea and send it out via the urine, which is where it got its name from. It's the major component, or the major reason for urine to exist, one of them at least. Now, some of the urea is not eliminated right away, and so it does actually cycle a little bit. This is also a nice summary of everything that can happen to dietary protein. To be fair, this is in cows, but it was such a nice flow chart that I decided to keep it anyways. So don't worry about like that stuff. Guess I should have just made my own. Anyway, it does show you some stuff. So anyway, basically the bottom line is that for catabolic processes, we're using this entire pathway of cellular respiration to get ATP. We're capable of making, through anabolic pathways, the larger molecules. Although for lipids and for carbohydrates, we're just making storage forms, whereas with protein, we're making proteins that are needed by the body. So that's kind of how we deal with our three major nutrients. And now we're going to look a little bit at metabolic state, which addresses all these processes we were just talking about, and just defines when they're being used. So the body exists in this dynamic state, right? We're always trying to maintain homeostasis, we're trying to maintain a dynamic equilibrium, but various things are changing all the time. So the body always has needs for nutrients, and it's going to draw on its storage pools in order to meet needs, and then it's going to add to the storage pools when it has excess. The proteins are a little different since we don't store amino acids, but we can break proteins down if we need to use amino acids for energy. Whether you're adding to storage pools or removing from storage pools depends on when you ate last. So we have two basic states in the body. We have the absorptive state, which is when you're actively digesting a meal, and we have the post-absorptive state when there's nothing in your small intestine to absorb nutrients from. So this is meant to demonstrate the various pools. So we have the polymers up here, proteins, glycogen, triglycerides, and then it's showing you how they can be converted to use to make ATP, and the various products that can come off of that, including our urea down here. So we call this interconversion, the ability for all this stuff to get moved back and forth between these three different categories to some extent, depending on the body's needs. So the absorptive state is also called the fed state because it's right after you eat. It lasts about 4 hours because that's how long it takes for the average meal to move its way through the small intestine and get digested and absorbed. So when you're in the absorptive state, you're going to have anabolism exceeding catabolism because you have all of the energy molecules that you need coming from your meal. And however many more you have than you need in the immediate moment, you will store. So we're making our storage molecules now, because we don't need to break them down. Now this is primarily regulated by insulin, and then supplemented by the parasympathetic nervous system. So insulin gets released when you eat a meal with sugar. So as long as your blood sugar increases, but amino acids will actually also trigger this to a certain extent, insulin gets released, and insulin increases the glucose transporters in most of your body tissues, encouraging them to absorb glucose. It also actually encourages them to absorb fatty acids and amino acids and store what they can or use the amino acids for proteins. So all of the nutrients are circulating in the blood from the digestive system. The liver is working very hard to store a lot of these things or to alter the triglycerides, for example, and send out VLDLs. Muscle cells actually get to use glucose, so this is their time to build up their glycogen stores. The liver is going to build up a glycogen store too. And the adipose is just kind of taking whatever excess adipose is available for storage as well. So it's all anabolic because we're storing everything we can and saving it up for when we're done processing this meal. Once this meal is done being absorbed, we enter the post-absorptive state. This is also called the fasting state. So the idea here is that now there's no longer nutrients coming from the gastrointestinal tract, so we have to rely on our body stores to supply our energy needs. The primary goal of the post-absorptive state is to maintain a normal blood glucose level because your neurons and your red blood cells need that. They can't use anything else. So it's mostly up to the liver to make sure that the blood sugar levels stay normal. So what we see is the liver is using glycogenolysis and gluconeogenesis. So breaking down glycogen and making new glucose from other molecules. Skeletal muscle can break their glycogen stores down as needed, and after that they're reliant on whatever the liver produces. Both the adipose tissue and the liver are breaking down lipids as well, so lipolysis and release of triglycerides and fatty acids into the blood for use by other tissues, including muscle. And then if needed, we will break down whatever the least important proteins are and use the amino acids as well. Now, most of this is, sorry, did I write it down on the next one? Apparently not. So, sorry. This is mostly mediated by glucagon and a lack of insulin. So without insulin, very few types of cells can pick glucose up out of the blood. It's pretty much just the brain, red blood cells, and I think the liver can do it too. So everything else in the body has to use lipids and proteins because we're doing what's called glucose sparing. We're making sure that all the blood glucose is reserved for the brain and the red blood cells, for the cells that cannot use other things. So the glucagon is stimulating the liver to do glycogenolysis and gluconeogenesis. And without the insulin, most of these other tissue types, they don't have the glucose transporters that allow them to bring glucose into the cell. Now, the brain and red blood cells in the liver, they always have, and I think the kidneys, they always have glucose transporters. They're not dependent on insulin, so that's why they're still able to use it or able to bring it into the cell. So now it kind of looks like this. Glucagon will get released if blood sugar levels drop below normal, and that's going to act on the liver to make more blood sugar and release it. It'll also stimulate adipose to release triglycerides. Muscle cells will start using their stores, and they'll be able to use triglycerides circulating in the blood, but they can't use glucose. Okay, so post-absorptive state. So usually we're kind of just alternating every few hours between these two things. There is definitely value in this post-absorptive state too. This is the ability to do this is where intermittent fasting comes from. Has some interesting studies behind it. Because the longer you stay in the post-absorptive state, the longer you're going to be doing this. And it seems to be beneficial, probably because this is how we evolved. We didn't used to have such ready access to food, and so post-absorptive states normally lasted longer than they do now. So here's another flow for controlling the post-absorptive state. So again, the trigger here is decreasing plasma glucose levels, increasing amino acid levels with low will also trigger this. So you'll get glucagon release if you eat a high protein meal that's got no sugar in it. So anyway, we get glucagon release, and we're gonna make sugar in the liver, release fat from the adipose. Once we increase blood glucose levels sufficiently, we shut off the glucagon release, and then the fatty acids are used by everybody else so that they don't have to use the glucose. There are, of course, other things that will help regulate this state. So hypoglycemia triggers a lot of things, including sympathetic activation. So we get the release of epinephrine, which promotes glycogenolysis and lipolysis in liver, skeletal muscle, and adipose. Sorry. We'll also get the release of other things like growth hormone, thyroxine. Well, thyroxine doesn't necessarily get released. Growth hormone gets released as well with low blood sugar because it does have the similar effects as glucagon. Thyroxine is going to influence our metabolic rate, as will sex hormones. And then while corticosteroids don't get released with low blood sugar necessarily, they do stimulate the same effects in the liver. So the gluconeogenesis and the glycogenolysis. All right, so that's kind of basic metabolic state stuff. Essentially, it just depends on where we're getting the sugar from, but we need to keep it at a relatively stable level so that our brains are getting enough fuel. Because remember, they're very metabolically active. The other thing that we look at here is energy and heat balance, because it's kind of all linked together. So energy balance is all about making sure that you have enough stores. We'll start with a little bit of basic thermodynamics, which says that energy can neither be created nor destroyed. All you can do is convert it. So when we're releasing energy from chemical bonds, and making ATP, we are using that energy for work. That work is usually anabolic stuff, but it can also be things like muscle activity, secretory activity, stuff like that. Any excess energy can certainly be stored, but none of these processes that we've talked about so far is perfect. They're all relatively inefficient. And what that means is that a lot of the energy that we're trying to use in these catabolic processes is actually lost as heat. So it's getting converted to heat, and that's a form that we can't necessarily use, except that we harness that to help us with thermoregulation and maintaining our body temperature. So this is super basic, but it's also overly simplified because it makes it look like it's very, very simple. So basically, people typically maintain their weight when the number of calories or energy that they're taking in is equal to the number of calories that they're burning. You'll lose weight if you're burning more calories than you're taking in, and you'll gain weight if you're taking in more calories than you're burning. But that being said, all of these other factors, hormones, activity levels, genetics, how old you are, your gender, all of these things come into play as far as like what's actually going on to control, especially how many calories you're using. So this makes it look very simple, but the reality is quite complex, which is why we have whole industries around this stuff. So the way that we know when we need to eat, it feels fairly straightforward most of the time. We're like, oh, I'm hungry, I should eat something. But it's actually a complex combination of nervous system and hormonal input. Most of it is mediated by that hypothalamus. The hypothalamus is capable of influencing eating behavior. We basically have a set of neurons that make us feel hungry and a set of neurons that make us feel full. And the hypothalamus knows which one to trigger or which one gets to be active based on a variety of stimuli. So we have stimuli from the digestive tract as far as the volume and composition of food. We have endocrine signals about the amount of body energy stores we have. This is mostly from the adipose. Other hormones like growth hormone, thyroxine, various other things. Body temperature can definitely have an influence. I don't know if you guys have felt the heat wave that we're currently experiencing, but that'll dull your appetite right there. And psychology is also going to have some effect on this as well. For example, we have very much like a culture, a lot of us get raised in America as like, you have to finish the food that you're given or the food that's on your plate. And we learn to ignore like the signals that tell us we're full, for example. And so that can kind of make it harder. When you look at it, it looks quite complex because it is, but basically in the short term, how much we eat, so meal to meal, it's mostly controlled by the hormones and the enteric nervous system signals coming from the digestive system. So what you're currently eating is going to influence how you're feeling. But when it comes to long-term controls, this is mostly about your hypothalamus regulating homeostasis of energy levels, as well as influence of insulin levels and leptin. So leptin is the hormone that is produced by your adipose tissue that lets your brain know how much adipose you have. It's basically the report about your energy stores. And based on these things, we're either going to trigger fullness or hunger. Another thing that has an influence here in determining how many calories you burn is your basal metabolic rate. So overall, your metabolic rate is defined as the total amount of heat produced by chemical reactions and mechanical work of the body. And that's because that's how we can measure it. The basal metabolic rate is essentially the energy you need to perform only the most essential activities, to keep you alive. And it has to be determined under highly standardized laboratory conditions where you're not eating, so you're fasted and you're relaxed, because we don't want stress to increase your metabolic rate when we're measuring it. So this is literally like the energy that you need to breathe, maintain your body temperature, and have your heart beating. Like, that's it. So obviously this isn't what we use, this is just what we start with. Things that can influence your BMR, because it is variable, there's no one standard BMR. Your age, typically the younger you are, the higher it is. Your gender, so males typically have a higher BMR for two reasons. One, because testosterone does that, and second, because the more muscle mass you have, the higher your resting metabolic rate to fuel those guys, and this is, again, thanks to testosterone. Body temperature will also have an influence, so the higher your core temperature, the higher your BMR, because enzymatic reactions happen more rapidly at higher temperatures. And then stress is, of course, going to influence this because you will increase your basal metabolic rate when you're stressed. It literally, you burn more energy when you're stressing out. Sadly, that doesn't always add up to you losing weight, as a lot of us probably know. Thyroxine, so thyroid hormone, is the most important hormonal factor in determining BMR. It's literally the hormone of your metabolic rate. So the level of thyroid hormone, in your blood is going to dictate the oxygen consumption and heat production levels of cells. Neurons are the only cells that are relatively impervious to that. They don't respond to thyroid hormone. And basically the way that thyroid hormone does this is higher levels of thyroid hormone can increase the amount of sodium-potassium pumps. This also means, because this using sodium-potassium pumps also generates a lot of heat, is why thyroid hormone is also a major factor in maintaining body temperature, which we'll talk about in a couple more slides. So basal metabolic rate is a baseline, but we obviously don't usually just lay around and breathe. So our total metabolic rate is how much energy we need for all of the things that we do. And again, it's all about heat generation. And I didn't say this before, and I should have. We measure that in calories. So the calories that we look at when we look at the nutritional value of our food, those are actually kilocalories. But it's the same measurement scale of heat generation. And you don't need to worry about it more than that. So total metabolic rate includes both voluntary and involuntary activities. Basal metabolic rate is the largest proportion of your total, pretty much no matter how active you are. However, in the short term, skeletal muscle activity does dramatically increase TMR. It's just really, really hard to be active enough all the time to massively crank it up. Interestingly, digesting a meal uses energy too. So even though we're getting energy out of a meal, we see an increase in total metabolic rate during food ingestion and digestion. And this includes all the processing that your liver does after the fact, because detoxification of substances that don't belong in your body can actually burn a lot of energy. So interesting there. Here's a little flow showing you the approximate breakdown of energy expenditure. So you can see that basal Using red on red is such a bad idea. Basal metabolic rate is just over 50%. This thermogenic digestion thing is not that much. And then everything else is the amount of exercise and then the amount of non-exercise activity thermogenesis. So non-exercise activity thermogenesis is everything that you do that is activity that is not actually exercise. I don't know if this one is accurate. I was just trying to find a good little picture. But basically anything, walking, cleaning, running chores, there's a lot of walking. Any of it that's not like actually exercise counts under this. Now, I personally would not count dancing. I think that's just exercise, but whatever. Like I said, this is not the end-all be-all of what NEAT means. This was just the cleanest, simplest picture I could find. So that takes us to thermoregulation, because all of this metabolic stuff does generate heat, but we can't just generate heat and be good. We have to actually regulate our body temperature. And we know this because we learned about thermoregulation as our classic negative feedback example at the beginning of class. So in order to maintain body temperature, we have to balance heat production, which is caused by all the things we just talked about, and then heat loss, right? And then obviously environmental temperature is going to have a major effect on this part. So heat production varies by metabolic activity of tissues. So the most metabolically active tissues produce the largest amounts of heat. When we're not physically active, your liver, heart, brain, kidneys, and endocrine organs are the most metabolically active. When you are active, it's your skeletal muscle and also your heart, but skeletal muscle by mass, I believe, is going to win over heart. So this is why this is the one we can change most rapidly. And of course, the regulation for this is in the hypothalamus, because the hypothalamus literally contains the thermostat of your body. It knows what it needs to do because thermoreceptors provide it with stimulus. So there's kind of a range of temperature. When something, or when you're in a thermoneutral environment, that means that the external temperature that you're in is doesn't require you to expend any additional energy to maintain your body temperature. So if it gets colder than that, then you have to expend energy to generate more heat and to not lose heat. And if it gets hotter than that, then you have to generate, or then you have to expend more energy to get rid of heat, which is ironic because then you're generating heat to get rid of heat, because energy expenditure means heat generation. Another thing that actually comes into play when it comes to thermoregulation is the fact that there isn't one universal temperature in your whole body. Give me 1 second. So we have a core temperature and then we have shell temperatures. So core temperature is the temperature of your internal organs, so your brain, and then everything in the thoracic and abdominal cavities. This is where your body temperature is typically the highest, and it's also where it's the most important. It needs to stay very precisely regulated and constant. It should not be fluctuating. It's also the hardest to measure, because you really have to get something in there to measure your core body temperature. Now your shell temperature refers to the skin and other peripheral tissues. This is typically the lowest temperature because either, well, no, it's just typically the lowest because we're trying not to lose heat most of the time. So we keep most of the heat in the core. And it's able to fluctuate a lot in order to help us maintain core temperature. So when we're hot and we're trying to lose heat to cool down, we can make our periphery very hot. But when it's cold and we're trying to not lose heat, then we can make our periphery much colder. So if we're measuring peripheral temperatures, they can fluctuate wildly and have no impact on our core temperature. The ways in which we can lose or potentially gain heat with the environment are these four. So heat exchange with the environment can happen in a couple of different ways. So radiation refers to basically energy from the sun, either directly or indirectly, or from other heat sources where we're literally radiating heat. So this would be in the form of electromagnetic energy, that's infrared. Conduction is transfer of heat by direct contact. So whenever you're actually touching something, you can gain or lose heat by conduction. This is why, you know, like lizards like to sunbathe. They bask on stones. And so not only are they getting radiative heat from the sun, but they're also getting conductive heat from whatever surface they're on to warm them up, because they don't internally thermoregulate as well as we do. So they have to use their environment more cleverly. The other two, so convection is where we exchange heat with the air. We can do this one of two ways. Usually we're losing heat because usually, I hope that you're in a place where the air is cooler than your body temperature, because otherwise it's really, really hot. Usually we're heating up the air closest to our bodies, and then the heat causes it to expand, it moves away, and then cooler air replaces it. So this is why we wear layers when it's cold outside, because that traps that warmer air next to our bodies and we're not constantly losing it and then losing more heat to the cooler air that's coming in. And then evaporation is where we lose heat when water absorbs heat. So liquid water on our skin absorbs heat and becomes vapor, and then that takes the heat away. We have both insensible and sensible evaporation. So what that means is that we have to lose water from our bodies in order for evaporative heat exchange to take place. Sometimes we do that on purpose. So when we sweat, that is, you know, deliberate loss of heat and water to get rid of some heat. But we also, nope, I've lost it. We add water to the air that we breathe in. Yeah, no, I can't remember the word right now. This is great. And so when we breathe it back out again, we're breathing out the water too, and we lose that heat. So that is an ongoing loss. We also lose a little bit of water through our skin, even when we're not sweating. And so that water is also evaporating. That's an ongoing heat loss too. So the ones that we're not really aware of in our breath and through our skin are insensible losses. And then when we're sweating, then that's a sensible loss because we're aware that we're losing that water. So thermoregulation takes advantage of the fact that the surface of our skin or the surface of our body is the part that's actually interacting with the environment through those four methods for heat loss and heat gain. And you know how this works already because we've gone over this thermoregulation stuff, but we just want to look at it real quick and see how we're tying it all together. So we know that when body temperature is too low, the hypothalamus gets informed, and it's going to make sure a couple of different things happen. So obviously we can shiver and use the activity of our skeletal muscles to generate more heat. We can also lose less heat by constricting blood flow to our periphery, so especially to our skin, We don't send as much blood to the surface so that we're not losing heat from that blood. The other thing, especially in the long term, if we're cold for long, is that our thyroid is going to release more thyroid hormone to encourage cells to increase their metabolic rate. As we use more ATP pumps, those sodium potassium ATPases, we are generating heat. This is called the thyroid's calorogenic effect. So burning ATP to generate heat. Now, of course, the opposite occurs when our body temperature is too high. Hypothalamus detects it, increases blood flow to our skin, and causes sweating. Both of these are going to help us lose heat by evaporation and convection. And then the thyroid will decrease its hormone production to decrease this heat generation. Okay, so that's thermoregulation in a nutshell. Last thing we're going to talk about is a little bit of nutrition, like just a teeny tiny bit, basically what it is and the two types of nutrients that we haven't talked about yet. So nutrition is the branch of science that deals with nutrients and nutrition, as well as the intake of food in relation to the body's dietary needs. These are a couple of good websites for nutrition if that's something you want to know more about. The basics are easy and the details are really complicated, just like a lot of other things. So we divide up nutrients into 6 classes, and we define them all as ingested chemicals used for growth, repair, or maintenance of the body. But we also think about it as anything we eat or drink. So water is actually the first and most important category of nutrient. It gets overlooked because it has no caloric value, but it's the most important nutrient. We have talked extensively about the next 3, and so I left them off: carbohydrates, lipids, and proteins. They are also called the macronutrients, along with water, because we consume them in large quantities. And the 3 that we've talked about are also the only ones that we can get energy from. The last 2 are the micronutrients because we consume them in much smaller quantities, but they're still quite important, and those are minerals and vitamins. The difference is that minerals are inorganic and vitamins are organic compounds. 1 second. One quick note about proteins before we go on to vitamins and minerals. Some amino acids we can synthesize in the body, and some of them we can't. So that gives us 2 categories of amino acids from a dietary perspective. The essential amino acids are the ones that are required in the diet. We need them in the diet because we cannot make them. And depending on exactly how we play this out, the numbers do vary, but generally it's somewhere in 9 to 10. Now that doesn't mean we don't need the other amino acids, because the more of the other amino acids we have, the lower our requirements for the required ones. But those are the non-essentials. Because technically speaking, if we didn't have them, we could still make proteins. But we kind of need all of them to make proteins in general. So I totally forgot. Last thing about proteins. Okay, so with the amino acids all being required to make proteins, we have a couple of different things that determine whether or not we actually make proteins out of our dietary amino acids or whether we use them for energy. First of all, we have the all or none rule, which is what we call the fact that we need all of our amino acids present to make a protein. If you're missing one, it doesn't matter how much of the other ones you have, you still can't cobble together the appropriate protein, right? Like you need the right sequence of amino acids. So if that's not happening, then we're burning them for energy because we have no storage form. The second thing is whether or not we're getting enough calories to use for energy from our carbohydrates and our lipids. If we are, then we can use our amino acids for proteins first. If not, we might need to use them for energy first. The third one is hormonal controls. So our hormonal state is going to determine how much anabolic stuff we're doing. Are we building our muscles and bones? Are we growing something where we need to make a bunch of proteins, or are we not? The other thing that we consider here is nitrogen balance. So I talked about the urea cycle before. The other side of this is nitrogen balance. So nitrogen balance is basically whether we're consuming enough nitrogen to balance what we have to lose or not. Protein is our primary source of nitrogen, so it's mostly about protein consumption. A positive nitrogen balance means that we are consuming more than we are actually excreting. And we typically see this in generalized anabolic states. So growth, physical training where we're adding muscle, or pregnancy where you're making a whole person, all of those things would be needing to ingest more protein than you're getting rid of. A negative nitrogen balance is when you're excreting more than you're ingesting. So we could see that, for instance, when we're relying on protein as our primary energy source because we don't have enough carbohydrates and fat. We can also see it occur in stress because prolonged stressors cause prolonged cortisol levels, and that will make us break down the proteins because our body is treating it as if we don't have enough energy sources. We want to be pretty much balanced most of the time. Okay, so those are the random little things about proteins. Minerals. So minerals are the inorganic elements required in the diet, and they have a variety of functions in the body. We've talked about the major players, and we're not really going to worry about the rest of them. So the majority of mineral in the body is calcium and phosphorus. Hey, I did not catch that typo. Because of the bone stores. And then when we look at the ionic forms, of course, the majority are sodium, chloride, and potassium. With vitamins, vitamins are the organic substances that we cannot derive calories from, and we only need them in small amounts. Now, some of these vitamins we can actually make ourselves, or we can consume in a other form and convert them. We divide them up into two categories based on whether they are soluble in water or soluble in fat. So the water-soluble vitamins are a bunch of B vitamins and vitamin C. Now, we don't store water-soluble vitamins, we pee out any excess, so we do need fairly regular consumption of these in our diet. Fat-soluble vitamins are A, D, E, and K, and we can store them with our lipid stores. We also need to eat lipids in order to absorb them because they're going to go in via the micelles, just like every other lipid-soluble thing. If you want to read an interesting article, this talks about how we figured out what vitamins were and how we ended up naming them for things. It's very, very weird. And so if you want a diverting little read, I do recommend it. So one last thing on the vitamins, because you're like, okay, what do we use them for if we don't get calories out of them? For a variety of things. So a lot of them aid enzymes in their functions. Some of them help in transferring energy. So in the cellular respiration that we talked about, there were two energy transfer molecules, FAD and NAD, and both of these are derived from B vitamins. So for even the most basic functions of life, we actually use some of these organic molecules. We've talked about vitamin D's function as a hormone in regulating calcium, and then some of them are used as antioxidants to neutralize various potentially damaging molecules that we generate as waste products. So vitamin C, vitamin E, and vitamin A are all antioxidants. And as we talked about in the nervous system, vitamin A is also very important in vision. That being said, as important as vitamins are and as much as we need good levels of them, most of us don't need to take vitamin supplements. So you shouldn't really take them unless prescribed by a doctor, because there is some amount of data suggesting that excessive supplementation, even if you're not overdosing, has been linked to like poor outcomes. So just because somebody's selling a multivitamin doesn't mean you should take it. That's my disclaimer. You can read that if you're curious. But generally speaking, most of us don't have deficiencies because a lot of our food is now supplemented, or what's the word? Supplemented with the vitamins that we need. If you're curious about vitamin and mineral supplements, here's a variety of websites. Quite interesting. And that's the whole lecture. So, anybody have any questions? Because that is all of our new material. On Monday, we will review the digestive system and metabolism. You'll have an exam on it the next day. And then you'll take your lab exam on the digestive system, and then we'll have a review for the final exam, and then you'll tackle the final. So in 2 weeks, we will be done. Just kind of crazy at this point. If you don't have any questions now, that's cool. Thanks for coming though. And please email me as usual. I am trying really hard to get everything graded, so I'm sorry that they're not all out yet, but everything takes time.
Are you still going to do the participation points?
I am trying. So that's kind of one of the things that those lab simulations are meant to do, is be your participation points.
All right.
Yeah, so obviously it's not going to be based on as much, but you'll get something for sure. And the first half of the class you did, so you'll still get points for all that too. Like it goes up at the end, because I just kind of track, like I was tracking attendance before, that all factors in. And now I'll just kind of look at that stuff to factor that in. So yeah, you'll still get some participation points.
All right.
All right, stop the recording.